In this video, we're going to talk about each of the organelles and what you need to know about them. And so this is the list of organelles, and we're just going to go line by line. This slide also, it's the same list, it's just copied and pasted. So this is the list of organelles that we need to understand. The first is mitochondria. Actually, you already know everything about mitochondria that you need to know. The mitochondria is the primary site for ATP synthesis. There's your mitochondria right over here. It's regarded as the powerhouse of the cell or the power plant of the cell. And you need to know, well, you already know that the citric acid cycle and the electron transport chain occur. They do their activities at the mitochondria. So without the mitochondria, can you still make ATP? You can. Remember, glycolysis makes a little bit. But mitochondria is the primary site of ATP synthesis, and this is where oxygen ends up being consumed. This part right over here regarding the two membranes, it's just a fun fact for us, so I don't necessarily need you to pay any attention to that. I just included it on there. The other interesting thing about mitochondria is that mitochondria actually has its own DNA. You can see written here, mitochondrial DNA. So the mitochondrial DNA is believed to come down through your maternal line, through mom's DNA. And yeah, it's its own DNA used to synthesize parts that are relevant or needed for mitochondrial function. So there you have it. Mitochondria, powerhouse of the cell, makes ATP. The next one then is ribosomes. So we talked about ribosomes, but let me give you a little bit more detail. So ribosomes, you already know, make proteins. You already know that ribosomes make proteins. They're involved in that translation process, which is reading messenger RNA to create a polypeptide chain, that protein. What I want you to see, though, is that ribosomes exist in one of two states. And the first is that sometimes ribosomes are just free-floating through the cytosol. So these little dots here, some of them are ribosomes. On the other hand, sometimes ribosomes are studded on top of this blue membrane that surrounds that nucleus. So this blue membrane that you see surrounding it is something called the endoplasmic reticulum. So unfortunately, I have to talk about the endoplasmic reticulum if I want to talk about ribosomes. So when you look at the endoplasmic reticulum, it's actually this entire blue structure. And part of that blue structure is studded with ribosomes, and part of it over here that I'm coloring in is not studded with ribosomes. It's bare. So again, the entire thing, this entire blue thing around here is the endoplasmic reticulum, and part of it has ribosomes studded on top of it, or in it, and the other part of it does not. The part with ribosomes is called the rough endoplasmic reticulum. The part without ribosomes is called the smooth endoplasmic reticulum. And if you go back to the list, you'll see that rough ER, endoplasmic reticulum, is listed here, and smooth ER we talk about separately because they have different functions. And so for right now, though, rough endoplasmic reticulum, we could say, makes protein. Okay, how does it do it? Because the rough ER, by definition, has ribosomes on it. But then on the other, so this slide is showing you these little purple dots or orange dots, whatever color that is, studded on top. So there's your rough ER, but again, some of these dots are also free-floating. So that looks like this. This is what we just said. The endoplasmic reticulum with ribosomes, that's what ER stands for, is the endoplasmic reticulum with ribosomes is rough, the rough ER. If there's no ribosomes, it's called smooth ER. So what does the rough ER do? It makes proteins. And I wrote here secretory proteins because you might ask the question, well, you know, what's the difference between ribosomes studded on top of the ER and ribosomes that are free-floating? For our purposes, let's say there's no real meaningful differences. I mean, the reality is some proteins tend to be made at the rough ER and other proteins tend to be made by free-floating ribosomes. But at the end of the day, for our purposes, both of them do the same thing, which is making proteins. It's also, the rough ER is also considered to be a membrane factory. So it makes different integral proteins. And by integral proteins, that word, I don't know what we used before, that just means proteins that are embedded within the cell membrane. And it also makes phospholipids. So these are all functions of the rough ER. The next slide is just giving you a little bit of a closer look at things. When you look at ribosomes, there's a smaller subunit and a larger subunit that need to hook up together to form this functional ribosome. I don't necessarily need you to know that information, but it's there if you're curious and interested. Again, it's simply the site of protein synthesis. I may ask you this to say, what do the ribosomes do? Do they A, do protein synthesis? That's true. Or do they B, do translation? Well, that's also true. So we'll find a way to make sure you understand what translation is. Okay, this is just another look at watching translation happen. There's no new information that you need to know on this slide, but if you're interested, go ahead and pause the video and take a look at that. So where we're at then, we talked about mitochondria, we talked about ribosomes, which can be free or embedded on the endoplasmic reticulum, which makes rough ER. All right, moving on then, Golgi apparatus. So the way I want you to think about it is that when you first produce proteins off the factory line through the ribosomes, they're pretty much almost ready to go, but they need kind of a final modification, final spit and shine kind of thing, and that occurs at the Golgi apparatus. So this entity here in green is the Golgi apparatus, and conveniently, of course, it's going to be located pretty near that rough ER. So what happens at the Golgi apparatus? Well, kind of the standard line is that it modifies, concentrates, packages proteins and some lipids that are made at the rough ER and that are destined for export. What do we mean by destined for export? It means that some of these proteins are destined to go out of the cell. So secretion, you can see here this term secretion by exocytosis, which we've talked about. And what do those proteins do? Well, some of them are plasma proteins. Some of them go off and talk to other cells. There's lots of other things that need to be done throughout the body. And so these are proteins and enzymes that end up being secreted. But again, the way you want to think about Golgi apparatus is that this is kind of a post-processing. After the initial manufacturing, this is post-processing. Not just post-processing, but then also following up from there, a distribution center. And so what I have down here is that if we think of ribosomes, whether free ribosomes or through the rough ER, if we think of them as a production center, producing what? Producing proteins, then the Golgi apparatus can be thought of as a distribution center. And so that is, again, these things that packages it into these little vesicles, these little bubbles, to go throughout parts of the cell or outside of the cell. Next then is smooth ER. So on our list then we did Golgi apparatus. Now let's talk about what you need to know about smooth ER. And again, this we've seen just a moment ago. Smooth ER is part of the endoplasmic reticulum that does not have ribosomes. So what does the smooth ER do? Smooth ER is involved with lipid metabolism, specifically cholesterol synthesis and lipoprotein synthesis. So the liver has tons and tons of, the liver cells, the hepatocytes have tons of these smooth ER because the liver is the primary site for cholesterol synthesis in your body. The lipoprotein, I know you don't exactly know what that is, and I don't necessarily want to get into it here because we'll look at that in the GI system, but you want to know that the smooth ER is the thing that generally handles lipids. Steroids are also, remember that steroids are derived from cholesterol, which seems to make sense because if I'm going to make cholesterol at the smooth ER, I might as well snip away at cholesterol a little bit to make some of these steroid byproducts. The smooth ER is also responsible for detoxification. So I wrote here drug and pesticide detoxification. Again, the liver has huge amounts of that. The kidneys have some as well too. Smooth ER is also responsible for calcium storage. Right now that seems like a weird thing. Why are we talking about calcium storage and not sodium and not potassium and not other things out there? Well, the reason is that calcium is commonly used as an within a cell, intracellular messenger. We'll see that very explicitly when we get into the muscle unit, because when we get in the muscle unit or module, we'll see that there's a specialized part of muscle cells, which is just smooth ER, but we give it its own name in muscles, and it's called the sarcoplasmic reticulum. And there we'll see the very critical role that calcium has to make muscles actually contract. If you don't have calcium, muscles don't contract. And so it's used as an intracellular messenger within muscle cells, but also in lots of other cells. So that is the smooth ER. The next on our list, let's see, we did smooth ER. Next are lysosomes and peroxisomes. So let's get through those. Lysosomes themselves, lysosomes and peroxisomes, we're going to clump together. I have them on separate slides here, but functionally they're clumped together because these are the two organelles in your body that break things down. They degrade things, they break things down. So lysosomes are famous for having digestive enzymes, and digest is just a fancy way to say break things down. You don't necessarily have to think of just food. But the digestive system is named the digestive system because you digest food. You break it down to smaller parts. But digest is a generic word that just means to break stuff down. So in the case of bacteria, if you get an infection of some kind or viruses or various other toxins, we may take those in by phagocytosis, which is a word that I threw out earlier with you guys, but we may take those in and then chop them up into tiny little pieces just to make sure they're really, really dead. And so digestive enzymes will do that, and these are housed within lysosomes. What else might you need to break down in your body? Well, old organelles. So all these things, it's not as if once you make them once, they're there forever. Sometimes you got to break things down and you got to regenerate new ones. But the digestion process needs to occur through, again, lysosomes. So what other things can you break down? Glycogen. I didn't use this term with you guys before with energy metabolism, but now is a good time to introduce it. Glycogen, you know, is the storage form of glucose. Glycogen, lysis. What is lysis? It's the breaking down. So when you store, let's say you eat a ton of spaghetti and you store away that extra glucose as glycogen, well, there's going to be times that you need to tap into that storage. So we break the glycogen down to release glucose, so then we can go ahead and do glycolysis. So that process is called glycogenolysis, and the lysosomal enzymes are the ones that tend to do that within those cells that store glycogen, which is liver and skeletal muscle primarily. So the lysosomes themselves have an acidic environment. I think previously when we talked about the first exam, I had said that, you know, your blood pH is about 7.2 to 7.4. It's ever so slightly alkaline. But then I did mention that, you know, some areas of the body are intentionally and deliberately acidic, like stomach when you want to break down food. But within a cell, the cell itself is generally a relatively neutral / slightly alkaline environment. But within the lysosome itself, if you were to crack that open, it's actually terribly acidic because the enzymes that are active in lysosomes really, really enjoy that acidic environment. Not to mention the fact that the acid itself assists in the breakdown process. And so things like phagocytes, which we'll look at when we look at tissues, phagocytes are immune cells that take things in by phagocytosis to break them up. As you can imagine, they have tons and tons of lysosomes because I got to break down these bacteria and other bad guys that I take in. So remember that lysosomes are this is what they do, and peroxisomes also break things down. But the way they break things down is a little bit different, and the stuff that they break down is a little bit different. And so peroxisomes are famous for having these two enzymes, oxidase and catalase enzymes. And you don't need to get too scared in terms of, oh my gosh, what does oxidase do and catalase? What does that do? But what I want you to see in the big picture is that these two things together, oxidase and catalase, these two things are going to detoxify things. They're going to detoxify things. And when you detoxify, oftentimes as a byproduct, including when you mess with free radicals, oftentimes hydrogen peroxide is produced, and hydrogen peroxide then needs to be handled because hydrogen peroxide is actually pretty toxic inside your body. And so it's interesting. What I'm really saying to you is this. Peroxisomes, what peroxisomes will do, they will take some bad things and make them even nastier. I know that sounds crazy, but just hear me out. Oxidase will take something bad, like alcohol, that we need to detoxify, and it makes them even nastier in the form of hydrogen peroxide, which will really mess you up. But after it makes the hydrogen peroxide, there's a step 2, which is catalase, which is going to neutralize that hydrogen peroxide and turn that into water. The reason I want to mention this is that sometimes you go from, in the elimination process, and we'll see this in the liver in Bio 142, the liver sometimes would take toxic things, it'll make them even more toxic and nasty just for a moment in time, just a very temporary process. And that's just part of the handling process so that we can eliminate it. Okay, but I say that to you because it leaves kind of room for imagination to say, oh, what would happen? Let's just call it a 2-step process. Step 1 makes a bad thing worse. Step 2 makes that worse thing clean and it gets eliminated. What would happen if step 2 broke down? If you only had step 1 of a detoxification process. And that's a legitimate concern for lots of folks out there with certain kind of liver disorders. In any case, that's why I wanted you to see this. And what do you need to know for peroxisomes? You need to know oxidase and catalase enzymes are there. There's a process that generates hydrogen peroxide and then eliminates hydrogen peroxide. Okay, other things that peroxisomes will do, peroxisomes will also break down fatty acids. So you've heard of this thing called beta oxidation. If you remember beta oxidation, it's where we break down fatty acids. Beta oxidation actually occurs in the mitochondria, but also occurs in peroxisomes. There's slightly different fatty acids that these guys will target. Peroxisomes also double up with smooth ER in the synthesis of cholesterol, and it can make these things called bile salts, which is important, again, later on in Bio 142. And again, tons and tons of these guys in liver and kidneys. All right, so that's lysosomes and peroxisomes. Again, together these guys are going to break things down. Lysosomes typically breaks down these types of items that I'm circling here. Peroxisomes then target different bad guys, but they use tools called oxidase and catalase, which lead to the production of hydrogen peroxide, which then we need to transform into water. This really is the main thing that I need you to know about peroxisomes. Okay, so where we're at is right over here, mitochondria, ribosomes, Golgi, smooth ER, lysosomes, and peroxisomes. And I think we'll cut this video here and we'll generate another one to finish out this list.